All right, welcome to Module 3 in our short course on risk assessment for food safety. So Module 3 is going to introduce you to some of the methodologies for estimating risk. So our learning objectives for this module are to understand top-down and bottom-up methods. I'll talk about what those mean. And then also to be somewhat familiar with the types of output that a risk assessment could create. So the two main approaches for risk assessment are top-down and bottom-up, and each of them serves a different purpose and uses different types of data. So either one of them might be relevant for the situation that you're in, depending on what you're trying to do. So I'll talk through both of them and give you some examples to help you think about which approach is really appropriate for a problem that you're working on. So the top-down approach looks backward from observed disease. So this really looks at data that you have on people that are sick and goes back to predict and model what percent of those illnesses came from foodborne contaminants. So there are, of course, a lot of gaps in this data, and there are methods to fill the gaps, as I'll talk about. And then this approach also, so the next approach is bottom-up, and in bottom-up, the analysis looks forward. So it goes from the existence of contaminated food, it might take it through the whole value chain, somehow to consumption, and then models the consumption and estimates, given that people are eating this contaminated food, how sick are people getting? So you can see that they use different types of data. So typically the bottom-up approach will focus on a very narrow part of the food system, so maybe a specific pathogen or a specific food group, and it might only focus on one part of the value chain. So digging a little bit deeper into a top-down risk assessment, so you start with some kind of public health surveillance that you have about people that have these negative health consequences. So whatever condition you're looking for, you would get the observed data on people that have illness, and you might do some modeling to say, well, these number of people were observed, but there's other people that are estimated based on the observed because of the data gaps. And so then modeling is done to account for that missing data, and then modeling is done to estimate the proportion of these illnesses from food. So typically, when you see country level estimates, they're going to be top-down estimates because it's just not realistic to be able to scale up to that kind of value. So these also might be referred to as system level estimates. And uncertainty ranges in these kinds of estimates is critical because of all the uncertainty in the data. So here's a infographic from the FERG data. So this is an example of the types of estimate that could be produced from a top-down risk assessment. So you might have seen this before. It's a summary of the data I mentioned earlier, and it's presented in different kinds of ways. So in another session, we're going to talk about, another module, we'll talk about different risk metrics. So this shows you an example of 3 different risk metrics. The first risk metric is a rate in which people get ill. So this was the likelihood I talked about earlier. They estimated 600 million people in the world, and that turns out to be 1 in 10 when they calculate it as a rate. Then the next metric they show is 33 million healthy life years lost. So life years lost is another kind of metric you can use. So it kind of adds information to just the number of people, but also talks about kind of how old they were when they died. And then 420,000 deaths, and that's the number we mentioned earlier. And then it's saying children account for 1/3 of these deaths. So there's a couple different metrics in here, and this is meant to show you an example of the kinds of information that could result from a top-down risk assessment. So there's 2 areas of data and evidence from a top-down risk assessment. Okay, I'm gonna keep going. So epidemiological links between symptoms and exposure, and then public health surveillance is the other type of data. And so one thing to keep in mind is that even in countries that have been collecting this data for a long time, there's a lot of data gaps here, and a lot of modeling has to be done to estimate the thing. So this is an example of the pyramid that we use to talk about our top-down estimates. So what we're trying to estimate is the exposures in the general population, which is all the way at the bottom of the pyramid. And then we're trying to estimate people that become ill because of that, because there are people that can be exposed that don't become ill. But what we have is the triangle at the top, which is the surveillance data, the number of people that were actually sick and that were confirmed sick from these particular pathogens. So you can see to move down the pyramid, you need to make some assumptions and do some modeling. So we go from surveillance to laboratory confirmed cases and to the lab testable organism. So there's some people that are that get laboratory confirmed, that are sick but don't have laboratory confirmed cases. So we have to model those. There's others where specimens are not obtained. So that goes down to the physician survey. And there's some in which people don't seek care. So each of these areas requires modeling to estimate the next level. So here's another way to look at it, that we have our total population at the bottom, and then we're really trying to estimate that second level when people become ill, but we only have the reported case up at the top, the yellow box, and we have to do risk modeling to estimate each of those other levels. So this top-down approach is really a public health surveillance-based approach. You need data on a couple of things. You need data on the rate of illness in the population, as we talked about, kind of who is ill, what have they been exposed to, and then hopefully you have some laboratory confirmed information on certain pathogens. And also I want to mention, you might have subpopulations of interest, because there are subpopulations such as the very young, the very old, pregnant women, and immunocompromised individuals who have different risks, different likelihoods and severities of harm from foodborne pathogens. So in addition to having information on the general population, you might also want to know that information if anyone has those characteristics, because you might want to analyze them separately. And then you also want to know what proportion of illness is attributed to food. So if there's a pathogen that could be in the soil, and could be in animals, and could be in water, and could be in food, in order to do this analysis to link the risk to food, you would need to have information on that proportion. So there's a couple of limitations in the top-down approach that are pretty significant, and each of these approaches gives you different kinds of information. So hopefully you'll be able to get a little information from whatever approach you take. The top-down approach only considers attribution to the point of consumption. So they're not looking at other parts of the process. They're really only looking at the food that was consumed. So there's really no evaluation of contamination within the value chain. So it doesn't really provide information on causal events, kind of what caused it. It does rely heavily on attribution information, as I mentioned, which has a great deal of uncertainty. So the second approach is a bottom-up approach, and this is probably a more traditional risk assessment that you might have seen. This really follows the contamination from the farm to the fork, so from the place of origin of the food all the way to where it's consumed. So it does include that whole value chain. So in this one, you need a lot more different kinds of data. You need concentrations of contamination at different points, the growth curves and log reductions when you're focusing on microbial pathogens, how much interventions reduce contamination. Then you want to know the relationship between the human illness and the product contamination. So how sick are people getting from what levels of contamination, consumption data, and production data. So here's an example of a bottom-up risk assessment, which is the Listeria one I mentioned earlier. Listeria, they call it in ready-to-eat foods, which also includes deli meats. And so you can see that over time, there was an estimate of how much illness was being caused by Listeria, and it had gone down a great deal, but there was still a lot of illnesses, and there was interest in, nothing had really changed since early 2000. So there's interest in trying to see kind of how to target better. So that risk assessment looked in detail at different types of ready-to-eat food and how they were and what their contamination level was and what kind of risk they were contributing. Because the truth is, if you have a food that has a high level of contamination, but it's not really consumed that much, it's not going to cause much illness. But if you have a food that a lot of people eat and only has a small level of contamination, it could make a lot of people sick. So the kind of intuitive part of the sense of how dangerous things are really can only be understood after a risk assessment is conducted. So here's a quick graphic that talks about, risk being in the center, and the top-down approach is taking you from diseases to understanding the risk of the consumed food, and the bottom-up approach is coming from the other side and looking at the raw materials and the processing and really building up that estimate of health impact. So in conclusion, in the review, in our learning objectives, hopefully now you have a sense of what top-down and bottom-up methods mean, and that you're familiar with some different kinds of output that a risk assessment could create. Thank you.